Bonjour à toutes et à tous, je m'appelle Guy Cochrane, je suis chercheur au laboratoire européen de biologie moléculaire. C'est avec grand plaisir que je participe au Forum de Paris sur la paix et que je présente notre plateforme. Hello everybody, I am Guy Cochrane from the European Molecular Biology Laboratory. It's a great pleasure to be participating in the Paris Peace Forum and to present our platform. So at the beginning of this year, we engaged in COVID-19 and became part of the scientific response to the emerging crisis. Our response was to provide organized data to the world to allow the scientific community to carry out research against the disease. So what I'm going to do over the next 20 minutes is tell the story so far of the platform that became known as the European COVID-19 Data Platform. I will talk about the data that we deal with. I will talk about the platform itself. I will highlight some of the enabling factors that have made it possible. I will talk about some of our early priorities and progress against those, and I will finish off with a brief look into the future. So the data that we focus on in the platform relate to biological molecules. So these are biomolecular data. The data cover everything from the sequences of the RNA and the DNA of the virus and the patients infected with the virus. It's about the genes and the genomes. We care about the viral genetic variation and the human genetic variation. It's about proteins, metabolic pathways, biochemical pathways, and including the compounds that can interrupt those pathways in medical situations. And then all of that is bound together by the scholarly literature, which really gives the scientific context for all these data types. So the reason that biological molecules are important and that biomolecular data are important in the fight against COVID is that they give us an understanding of the disease. So with biological data, we can understand the biology of the virus, the biology of the infection process, and the biology of the human response to that infection process. And we can also use the genetic variation of the virus to identify and track. So we can discover transmission events between individuals, between communities, and from country to country and around the world. So with that layer of understanding of COVID-19, we then allow the development of effective targeted treatments. We can enable public health interventions to be designed and deployed, and we can enable the design and the production of vaccines. And in a very elegant circle, because molecular methods are used to assess the effectiveness of these different interventions, we end up producing, as we go along, more data related to the biological molecules that better inform and refine our understanding. And so it's a virtuous circle. So the European COVID-19 Data Platform is shown on the left here. We were able to make a very rapid start. We started the technical work in March and we launched in April. On the right-hand side, you see the launch event with the President of the European Commission. The platform on the left is as it was at launch. We had already significant volumes of data of different types, and we had a number of tools. But since launch, we continue with an ongoing, very energetic program to add data and to add tools and functions. So every day, new data will appear in the platform, and every few weeks, there'll be new functions and new tools. And the reason we're able to make such a rapid start was because we have very strong foundations to build upon. And so the European Bioinformatics Institute, which is my part of the European Molecular Biology Laboratory, has for many decades been the point in the world in which scientists deposit their biomolecular data, and we curate and organize and then present this back for use and reuse into the future to the scientific community. Elixir on the bottom left is the European coordinating network for biomolecular data in Europe. And so there, partnering with Elixir gives us immediate access to a network of experts in dealing with these data. On the top right, we have the European Open Science Cloud, who bring in expertise in technology, in data standards, and in infrastructure, and also provide an entry point and an access point that we can use to connect with many other scientific data infrastructures. I'll come back to that. And then at the bottom on the right, we have, over the last 8 years, collaborated with a number of important medical and scientific academic groups in different institutions on the COMPARE project. And specifically here, we've built up a layer of infectious disease specific database, services, tools, infrastructure to support people doing infectious disease work, sitting on top of the broader infrastructures provided by the other three. And so here we have the partners Erasmus Medical Center, RIVM, in the Netherlands, the Danish Technical University, the University Clinic in Heidelberg, Germany, and the Eötvös-Loránd University in Hungary. And our platform work is supported by the Horizon 2020 program of the European Union in the project shown on the right. So the platform itself, our aim is to make available relevant biomolecular data relating to COVID-19. We make these data openly available and freely through simple interfaces and flexible interfaces. It's a diversity of data of the different biomolecular types, and we aim for integration so one can navigate from one data type to another in sensible ways because the data very typically connect. We want to enable rapid sharing. It's important in the fight against COVID that data become available very rapidly as soon as they're produced and validated. They need to be available for the scientific world to use them. And we provide a global service. So the content, the coverage of the system is global. But we have it's presented globally, so people can use it and do use it from around the world. So the kind of data flow that one imagines, the process that sits around this, I will step back just to cover three of the core technical components. On the left, we have the SARS-CoV-2 data hubs. So this is a toolbox that allows people dealing with viral data to manage, process, analyze, and interpret their data, and then push them into the public collection, the centralized collection of all viral sequences. In the middle, we have a database that is federated. So this deals with the sensitive human data. For privacy reasons, these data typically don't leave the country in which they're produced. They're kept within national infrastructure under national GDPR regulations. Yet we need a single technical system that allows them to be connected such that one can search across the whole data set, across all of the nodes within the network. And one can apply the same interfaces. For example, if I'm making an access request, it's useful that that access request process is the same across, regardless of where the data live in the federation. And then the third component on the right is the COVID-19 data portal. And this is really the entry point for most of the people using the system. This brings together the viral data, the sensitive human data, but many other data types. And I'll show these in a moment. And this is really the way in which most people using the platform engage with it. And so I'm just showing here the process that around those components. We have data produced typically where the patient is, in the hospital, in the clinic. Those data are organized already into national health and public health databases within the different systems that exist. And then we've organized and choreographed these flows and these referencing of different types of data into the different components. So here we have the viral data, here the sensitive human data, for example. And then the portal at the end of it really presents a view on that. It's a window onto all these different databases that allows an organized entry point. We serve this, as I said, openly to the users, be they from academia, from industry, from government, from health, from public health, the medical world, and so on. So the focus of the platform, the center point is really the biomolecular data. However, for many studies, in order to make sense of those data, one needs to understand the context surrounding those data. And so it's particularly important, for example, to understand clinical conditions, symptoms, clinical measurements on patients, to understand diagnostics, to understand the therapy, the history of therapies that are given to a particular patient, the exposures to previous disease, the contacts that the patient may have had, and then going out even more broadly to look at the social sciences, so behavioral data, looking at socioeconomic data, looking at population epidemiology and social media content. And so really we need to have a system that, while focusing on these biomolecular data, it presents this in the broadest context. And in order to achieve that, what we have is a system that has many layers. So some parts are centralized. The core molecular data can be centralized, and we have those over in the European Molecular Biology Laboratory. A lot of this exists nationally. So the sensitive human data, including the biomolecular data, but also the clinical and the public health data, tend to be organized within national infrastructure, and we need to be connected and fluid with those. And then at the European level, we have many infrastructures that do a very good job already of organizing data types of particular importance, albeit outside biomolecular data. So for example, compound screening data, clinical trials data, biobanking data, imaging data, and these are of importance to COVID-19, and our system allows the connection. And then at the global international level, we have operations to share data with systems around the world and to work on standards and to work on the conventions for doing that data sharing as well. So I just come to two of the important enablers of the platform, two factors that have really lubricated the system and allowed us to set up quickly as well. And the first is collaboration. To build a system of that complexity quickly, it's actually been a real joy to be able to set up collaborations and deliver things through those collaborations so quickly over the last few months. And this is just one of the examples. And we now have a number of countries here who are providing national data portals, national COVID-19 data portals. And these serve to allow the scientists within different countries to come in and navigate what's provided nationally, so the support, the data management services, but also the national studies, perhaps those sensitive human data studies, the cohort studies, and so on, to manage access to all of that, but then to allow the user smoothly to move across to the central data portal for those elements of the system that are provided centrally, such as the viral data. And so this is a really, really important aspect of coordinating and presenting the users with a sensible entry point for people in different countries, but in an effective and efficient way that connects everything together. So the second factor, the second lubricant of the system that's enabled things has been openness. And I think we enjoy, in the world of molecular biology, we enjoy a history of very open sharing of data. And time and again, this has been really useful in allowing our science to advance very quickly. And so the platform really builds on that. It leverages it and it builds upon it. And so having open data means that one can very quickly connect different data sets and communicate different data sets. And so it becomes very practical to be able to build these kinds of platform. Of course, there are sensitivities, and one has to protect the privacy of research subjects, the patients for some types of data. But we can build systems that are open enough to allow research, even though they're respectful fully of patient privacy. And so I'm including this concept of openness, those kinds of data where one does need some kind of protection. It's not just data, it's also software. So we build and we take advantage of other people's open source software, open data standards. And so we also contribute and build data standards, and we use those data standards that are produced openly by other groups as well. And I'm just showing here only a selection of the different standards and data exchange agreements that play a part in the European COVID-19 data platform. There are many more, but many of these are organizations that for many years have been working on open data, open software, and open data standards. So how are we doing against the early priorities? So I have two priorities to talk about. The first one was that we really wanted to make all of the relevant data relevant to COVID-19 available in one system and easily navigable. And so this is where we stand with that. We have something like, if you add these up, 400,000 plus different scientific records that have a relationship to COVID-19. So we have an important resource here. And this is bringing in many of those different data types that I mentioned right at the beginning. So here we have sequencing. And if you look at the numbers, these are some significant numbers. I'll come back to the raw reads. This is human sequencing, proteins, expression, other types of data, this is biochemistry, and then the literature. And if you go and look at these every few days, you see these numbers changing, and particularly fast changes are the sequences. People are doing a lot of sequencing and putting it in the platform, and people are writing a lot of papers in the scientific literature, and these are appearing in the platform. So these are growing quickly. If you drill down, the platform allows you, the portal allows you to go and find these data types. So here, for example, you're looking at you just about see some of the images of protein structures, for example. So the second priority was really to make sure that the world had access to precision, high quality, trustable genetic variation from the virus. So the variation, as you will remember, is important for understanding the flow of the virus around the world, but it's also important for understanding the biology of the virus. Accurately understanding variation as it appears is really, in the virus, is really important. And in order to do that, because sequencing technology produces errors, it's not a perfect system, one has to go through a process of separating out the noise from the system from the genuine genetic variation. And the way to do that is to treat all of the data systematically. But at the very beginning of this, back in March, there was very little sharing of any forms of raw data that are needed to be able to do that systematic processing. And so we set about fixing this. We had a significant effort to enabling people to put their data, to generate their data, put their data into the system. And we had a campaign that went country by country to help people to do that. And there was a lot of support for compliance with standards and curation and so on, and quality control. You can see that this has been effective. On the top right there, you see the growth of these data sets. We now have 125,000 raw data sets from sequence viral isolates coming from 47 countries. The map on the bottom right shows those different countries. All of these are in the platform. You'll notice, you can just about see in red, these have come directly through the data hubs system. In blue, they've come from other sources. But the data hub, so the platform itself has through this effort, has made effectively 3/4 of the data. They put 3/4 of the data available to the world into the system. So it's been a very important effort in mobilizing these data. And indeed, we now have enough to be able to do systematic analysis, and we're doing that, and we will be, over the next weeks and months, rolling out some of these analyses as appropriate through the platform. And this is just one of the windows onto how one can navigate through those analyses. So I've mentioned the progress against those two early priorities that we set for ourselves, but of course at any one time there are many different things happening, and I can't represent them all for lack of time. But here I just highlight a selection of things that are happening that I could have gone into more detail on. So on the top left we have work on the federated database system that I mentioned for sensitive human data, building out different nodes within different countries, and we're expecting to see some of the European countries produce these before the end of the year. On the top in the middle, we have cohort study support. So these are tools to allow patient cohort data to be better organized and better discoverable through the system. Top right, we have at any one time many different analysis tools of different types targeting different data types being developed. Bottom left, not all of the data that we deal with have the appropriate data standards, and so sometimes we have to develop them. So this is an effort on the serology, serotyping data side, seroprevalence side. Bottom middle, promoting openness in data and promoting fair practice where one makes one's data discoverable and reusable. We have an active role in pushing for open data around infectious disease. And then bottom right, we have various activities around data curation and quality control to make sure that the data that we're serving are as useful as they can be. So I'm going to finish just to show some examples. It's an open system. People don't need to log in to get to it. They come and they look at data and they navigate the data. So we don't track how people are using exactly, but we know about anecdotes. So I've got time just for 3 here. So on the top left, we have drug development, and this is from the Open Targets group. And so what they did is they accessed data that are available from the platform. They ran analytical processes and curation processes, and they were able to prioritize different targets, so different gene targets that may be used for therapy against COVID-19. And they were able to classify the different compounds that have activity or show promise of activity against those targets, and to classify them according to their status in different phases of clinical trial, for example. And so this is a really important catalog for people in the drug discovery and drug development world that's based on the data from the platform. This one's actually come full circle. So we now push that back into the platform, and all of these data are now available from the platform directly as well. On the bottom in the middle there, we have an informatics usage. This is from the Galaxy project from Belgium and Germany. Galaxy is a cloud-based platform for biomolecular data processing and analysis. And what the team have done is built a whole set of analytical tools that allow people to customize analysis of viral data from the platform, and to manage those data and to push things back into the platform. And this is a layer of interface for users that sits on top of the platform, so it pushes and pulls data to and from the platform. Then the third one, top right, is a complex case where the platform is being used. It's early days, but it's being used to connect different data types. So in the Netherlands, there is a very rich history of collection of different data relating to infectious disease, deep viral data, deep host genomics, transcriptomics data, many laboratory data and public health data, but these all live in quite fragmented systems in different laboratories and in different parts of the national system. And so the platform here is helping to connect all of these together to allow complex analyses that need to cross these different data types. So finally, I just want to consider the outlook, and the immediate future is sort of laid out. We continue with the extensions of the platform to make it more usable, to add tools, to add functions, and certainly to add more data, and to support more people in using it. We, at some point in the future, the crisis will lessen, and we hope come to an end. But at that point, we'll have a very rich and very comprehensive resource that we can use for retrospective work. It will be very important to understand how the different interventions that were made around the world, how they actually played out, and how the data could have been used or was used effectively to govern those kind of decisions. And so having this complete system, even after the crisis, will actually be a really important asset for the future to understand the state of our pandemic response systems. We've learned a great deal about collaborating around infectious disease. I've mentioned these lubricators, these enablers, open data and broad collaboration. And a lot of it's been about communication at all these different levels. And that's been really important. And that, of course, will always be important. We know to move quickly as one needs to, one has to reuse, extend, adapt, and reconfigure existing components. One can't build a new system. There's no need to build a new system. These components, when fitted together, work very well. To do that, technical standards are really important. So you can have modular systems where the components can talk to each other technically. So I think it's almost certain that what we've done has a relevance for the next outbreak or the next pathogen that challenges the world. But I think in more peaceful times, there's a relevance in surveillance. So using some of these systems to track before you have outbreaks, or when outbreaks are only minor, to track infectious disease around the world. But I think from some of the connections we've made into the clinical world, I think also there are some possible applications in the future, more generally in health research, perhaps outside infectious disease. So there I leave the presentation and I turn to some of the questions. I'm just trying to see where these questions are. Okay. Okay, so I can see the start of the questions just appearing now. So how can countries struggling to collect their own data and research benefit from the platform? So I think for those countries that have programs to generate data, which I think now is probably most countries, then we have the platform provides a whole host of tools that can be used to support different parts of that process. If we take the viral sequence, where we provided very deep support, that's everything from actually advice on sequencing methods and how to make sequencing libraries, but the focus is more downstream on the analysis and the data management. And so we have a whole range of different tools that we can provide to people to manage their data locally, or to use our system to manage their data. They can do this prior to sharing their data, and then to run different analytical processes and to start to interpret. And that's true for many of the different data types that we have tools specifically that people can use. Sometimes they use those tools locally and they install them locally. Sometimes they use those tools remotely, so they connect to the things we're offering more centrally. But around all of that, there's a significant support effort. So we have an email help desk, and we have calls with people who wish to use parts of the system. So we have a significant capacity to help people to push data into the system. And the pushing the data into the system is sort of goes hand in hand with doing research, because one needs to understand what's happening nationally in the context of what's happening in neighboring countries, for example. And so that process to put data in, the conversation and the support around that is the same as the support and the process around discussing how one can do analysis and access things from the portal. So the next question which challenges did you face in building this data platform? Well, I mean, the big thing I think was that we had to move really quickly. We had a really good head start from the infrastructure providers and from the work we've done with our partners, but we needed to be operating a system that would present an interface to users within months. And normally these projects take a significantly longer time to start things working. So it was challenging to get people very quickly to switch to focusing on this. We're very fortunate at the European Bioinformatics Institute, where we have a lot of very expert bioinformaticians, so we could redeploy and second people. And of course, the collaborations externally were absolutely essential. But of course, setting up collaborations fully remotely, because of course all the participants were locked down at the time back in March. Setting up these collaborations fully remotely is not straightforward, and that was a major part of the work. But I think it was delightfully successful, actually, that we were able to do that. So I'm looking for the next question. What has been learned with this platform for future pandemics? Do you plan to develop it beyond the COVID-19 crisis? Yes, so I think we certainly do plan to continue to develop it. I think, as I mentioned, it's still on the slide here, that actually this will be an important resource for people to assess how we performed in fighting the pandemic this time round. But I think then we know a lot more about how we can rapidly deploy in future outbreak scenarios, in future pandemic scenarios, where we can much more rapidly deploy the components that are really essential. And I think then in those peace times, we will be able to use these components to do more routine surveillance. I think there are opportunities with some of these components to redeploy them into areas, perhaps going beyond infectious disease. Okay. So the next question, how can people access information on how data are used? So this is a challenging question. It's a good question. The convention is that when people use, so the data are open, and we don't ask people how they're going to use the data when they come to pull the data. The convention in the open data world is that one, and certainly in the scientific world, is that one cites the source of the data. So if I do an analysis on somebody else's data set, the convention is that I cite I reference and I cite that original data set. And we have systems, technical systems of accession numbers and referencing systems and permanent pointers to data and data collections that allow people to do this. So if I publish something in the literature, then conventionally I would cite the data that I've used, and then one would be able to go to the platform and find those data. And so rather than having a system that points forward and tries to track how things happen downstream, we have a system where people cite their sources and then you can reconstruct the pathway. But the answer is that we don't track. We don't try to, we like to have stories and we like to see people are using things, and we certainly like to have feedback on how people are using things. But we don't directly track exactly how every data set is used from the platform. And I think probably the final question now, is there room for improvement? How can the platform be improved? Well, yes, of course. There are many tools that don't exist in there. The viral data that I mentioned, for example, we don't yet provide the full analysis. In time, we want to provide a stream of analysis on the incoming data. There are many visualization tools that we're working on for visualizing data, for doing phylogenetics, many different analyses that we would like to support. And so we have great plans to do these things, and we look forward to doing them. But certainly there is probably more to do than we've already done in the last few months. So I think there I have to close it. I'm getting a note to say that we only have a minute left. So I thank you very much for your attention and for your questions, and I say goodbye. Je vous remercie de vos questions et de votre attention et je dis au revoir.